It's a perfect introduction. Thank you so much. Thank you especially to Dr. Farah for inviting me. I'm excited to be here as an alumna. I'm excited to be here talking to people outside of my immediate niche. So this is a really cool opportunity for me to share my work more broadly. Hopefully it's accessible to everyone here. And it's also an honor to be speaking in a more public forum, because I know that Dr. Farah is such a good public neuroscience advocate and speaker. So it's an honor to receive this invitation. And I will say, the course I took at Penn, I thought that I was interested in poli-sci, and I took a course on law, ethics, and neuroscience, where Dr. Farah taught the neuroscience portion, and that changed my life, and here I am. Yeah. It was great. So my lab studies how early life stress changes the course of brain development itself and increases risk for depression, anxiety, and other psychiatric disease. By the same token, we're interested in understanding how early life stress promotes these risks, and then how we might also be able to reverse those risks from a neuroscience perspective. So I'll take you through today what we know from a lot of the human literature that motivates our animal studies, as Dr. Farah said, and then our findings within the brain, really focusing on a cellular and molecular level. And hopefully bring it back out to how we can use this information to understand, again, us as humans. Okay, so first, what is early life stress or childhood adversity? Normally when we say something like this, we think of abuse, and this can include physical, psychological, or sexual abuse. Many people may also think of neglect, and abuse and neglect, unfortunately, are experienced by about 1% to 2% of children in the United States annually. Early adverse experiences or adverse childhood experiences, these ACEs, which I'll talk about, also include exposure to household dysfunction. You can see some examples of that here: violence in the home, substance use by parents, potentially divorce as well. And I'll come back to this, but if we include household dysfunction, this actually impacts about 2/3 of the children in the United States. Childhood adversity can also encompass sort of larger, more structural issues, low socioeconomic status, such as poverty, which affects about 20% of children, and systemic internalized racism is a huge childhood adversity that many children experience, unfortunately. Childhood adversity can also encompass loss of a caregiver and or home. Incarceration affects of a parent affects about 7% of children in the United States. And loss of a parent can also be due to death. Unfortunately, in the last couple of years, this has brought a really big focus from the pandemic, where more than 140,000 children have lost a primary, secondary caregiver to COVID death. And this was a statistic in October, and as we know, unfortunately, we've surpassed this number. About 3% of children in the United States also experience homelessness. This is obviously an extreme form of childhood adversity as well. And this can be due to a number of factors, including just displacement from natural disaster or violence in the home. But childhood adversity doesn't even begin to describe it for millions of children worldwide. Children are disproportionately affected by violence and displacement, and IRC and UNICEF estimate that about 30 million children worldwide have fled conflict and crisis, such that the number of child refugees has actually doubled in the last 10 years. Migrating children are at the highest risk for harm, abuse, deprivation, abduction, malnourishment, and other extreme stresses. This is, again, an older statistic. I think this was as of 2018, but the United States had more than 10,000 migrating children in detention facilities. And unfortunately, over the last week, this has been put into stark focus. This is a global problem right now. These are images that I took from Twitter over the last week, where children are being affected by violence and displacement in their home country. If they're not leaving, as depicted here, they're staying in uncertain conditions. So a better understanding of how early adversity alters brain development and increases the risks that I'll talk to you about in a minute really is urgently needed. So I told you that sort of the more common ones we think of, or the less stressful versions of childhood adversity are quite common. About 2/3 of children will experience some kind of adverse childhood experience, or ACE. And if we want to visualize this, the children in no color here are ones who may not have experienced any childhood stress, but you can see the majority of children have experienced something, with warmer colors indicating more stressful exposures. So what does this do? What effect does this have? As we increase the number of adverse childhood experiences, we increase risk for a lot of things. My lab was particularly interested in about a 4-fold risk for anxiety and depression. There's also an elevated risk for substance use across the lifespan. Children who've experienced at least 4 ACEs have a 30-fold higher likelihood of attempting suicide at some point. They're 7.5 times more likely to become a victim of violence. There's also increased risk for physical health. It's not just about mental health. 2 to 3 times the risk for cancer, cardiovascular disease, diabetes, obesity, and other diseases, which adds to a 20-year difference in life expectancy without intervention. While there is a lot of plasticity in brain development that can lead to these long-lasting effects, we can also hopefully take advantage of the plasticity in childhood development if we do intervene in time and reset these courses. So one thing I'm particularly interested in is in this increased risk for anxiety and depression. This is illustrated by a study about 10 years ago now, where even in the absence of any past year major stressful events, so 0 stressful events, children who've experienced at least 3 adverse childhood experiences have an increased prevalence of major depressive episodes and anxiety disorders. You can see these top little marks here. However, we're really interested in trying to understand what happens in the brain when we have a combination of early childhood adversity and recent stress. And it seems like early childhood adversity sensitizes an individual's response to stress later in life. So if you've also experienced recent stress, your likelihood of major depression goes way up. This is a synergistic interaction. So we're trying to understand what happens in the brain. I will pause here and also sort of take you on a side project that I was fortunate enough to be to help lead, actually. When I first started my lab in 2019, this was before COVID, I had an incredibly talented undergraduate pre-med but Spanish major join the lab. And the mouse work that we do, the type of early life stress that we impose, which I'll tell you about soon, is maternal separation. And before the pandemic, our news cycle was actually really focused on family separation of migrant children at the border. And so before I knew it, she went to the Spanish department and got permission and funding to go down to the Texas border in the summer of 2019 to try to understand what is the impact of childhood trauma before migration, experience in U.S. immigration detention facilities, and forced separation among asylum-seeking Spanish-speaking children. So she ended up conducting interviews with parents of 84 migrating children. Unsurprisingly, perhaps, pre-migration trauma, so trauma experienced before they attempted migration, was positively correlated with symptoms of PTSD. This supports vast literature. However, the second strongest predictor of PTSD symptomatology was the length of separation among the children that had experienced separation. Luckily, for better or worse, the news was all about these really long, dramatic separation periods. Children had been separated for months from their parents. You saw these images of children not even recognizing parents when they were reunited. We did not have children like that in our sample. This was a limited sample. But one thing we saw was that children who had experienced at least a moderate level of pre-migration trauma did seem to have this increased sensitivity to the stress of parent-child separation. So it's not statistically robust because it was a limited sample size, but we do think that this supports the finding in this vulnerable population of children that there's a sensitization to the stress of being separated from the parent. Obviously, we hope that this can support policy changes so we don't enact further trauma on these children. And this should also inform how other nations and we treat the children who are displaced in the current violence across the world. Okay, so we are interested in what's happening in the brain following these types of stress. I'll give you some background on what we know from human brain imaging, and then I'll take you through work in my lab using mouse models to uncover molecular and cellular effects. First, focusing on circuit activity changes, so the cells in the brain and how their activity changes in response to early life stress and adult stress. Diving into those cells, looking at how genes are expressed, and then how those changes in gene expression is regulated by the epigenome. And then a few slides at the end talking about promoting resilience and lessons from both our molecular studies and from psychology. So what happens in the brain following early life stress? In order to understand this, we also need to understand what's happening during brain development more broadly. Early in development, brain development is all about developing our sensory systems, so vision, hearing, touch, etc. As we grow, we are developing systems related to symbols and ideas, social relationships, language, attention. Later, increasing our cognitive abilities, making predictions, learning math, reading skills, a sense of self and others. And then adding on additional skills, critical thinking, judgment, impulse control, and decision making. On the more neuroscience side of things, myelination is rapidly occurring throughout childhood. We're forming synapses and ultimately refining synapses. And so what happens when we have trauma? These are the expected systems that are developing. What happens when we experience early life stress of some sort? And there's a lot of hypotheses that maybe we're shifting the course of development itself, either accelerating development or blunting development. And it turns out that in different brain regions, we have evidence of all of these different trajectories happening. For example, in parts of the brain that are sort of thought to form a response to stress or threat, including the amygdala, there's normally a reduced connectivity between this region of the amygdala and the prefrontal cortex for decision making across development over time. So there's a normal decrease in connectivity between these regions. And this is actually accelerated in children who have experienced early life stress. So stress will activate this brain region. The connections, as I just said, thin normally from childhood to adolescence, and this thinning is faster with childhood adversity. So these changes in the actual trajectory of brain development itself. There's a lot of evidence from the hippocampus. So this region of the brain is involved in learning and memory, where poverty and other childhood adversity reduces the size of the hippocampus, reduces the number of new neurons that are made. And there are changes in many directions in the connections between hippocampus and other brain regions, and difficulty with memory formation and recall in children who've experienced a lot of early life stress. So a lot of research has been focused on the amygdala and on the hippocampus. My lab is actually very interested in reward circuitry in the brain. So we call it reward circuitry, but these regions, including the nucleus accumbens and ventral tegmental area, which I'll talk to you a bit more about, also are involved in motivated behavior more broadly and response to stress. What we see in these brain reward regions are blunted development of brain reward circuitry, decreased circuit activity during reward anticipation, reduced activation of brain reward regions, and these also predict symptoms of anxiety and depression. Work by members of our audience. Okay, so how can we use this to inform? Well, this is a lot of changes happening at broad levels, at a level of activity changes that we can detect with neural imaging. And what I'm really interested in is really diving into the cellular and the molecular level. And we really can't do that in human brains as well. So we're using a mouse model. So I'll tell you about our mouse model that we're using, and then take you through some of the changes in the brain that we see at the level of cells, at the level of gene expression. This is a heat map where yellow is increases and blue is decreases. And I'll keep coming back to this in the level of mRNA of genes, and then how those changes are regulated by the epigenome. Okay, so first, how do we model early life stress in a mouse? We actually use a combination of maternal separation, so we're moving pups away from the mother for about 4 hours a day, and then they get reunited with their mother, unlike what we're doing at the border. And we take away some of their resources in the home cage they live in. So normally, mice get to make this nice, cushy nest. It's big and fluffy. They hide there. It's very comfortable, and they keep all the pups in the nest. And the stress that we give to our stress animals, we take away some of that bedding material, and it makes the moms a little bit frantic. They're off the nest a lot more, and they're kind of more disorganized behavior around their pups when their pups are there. So we're sort of throwing all this at them. And we also tested 2 different periods, or potential sensitive periods, in a mouse development. I'm calling ELS1 and ELS2. The first period is right after birth, and the second period's shifted a little bit. Many people do early life stress research in rodents right after birth, sort of thinking birth is birth, but there's actually some evidence that that rodents are not able to mount a full stress response until a little bit later. So we're going to test that hypothesis. And then because we're very interested in how early life stress might increase susceptibility to future stress later in life, we put half of our animals through adult stress. And the adult stress that we're using for the most part is social defeat stress. So this is essentially being bullied for 10 days in a row. We have our experimental mouse. It goes into a cage territorialized by this big, aggressive mouse. They actually fight for about 5 minutes, and then we separate them across a semi-permeable barrier. So they're no longer fighting, but they have all this sensory contact with the aggressor mouse, and it's trying to sort of get at them for the rest of the day. So this really think is more of a psychosocial stressor for the rest of the day. And they see a new aggressor mouse every day for 10 days. One of the main behaviors we're looking at is how they respond socially. So we're looking at social avoidance behavior. This really is a robust, reproducible, and actually bidirectionally modifiable sort of endophenotype that we think we can model in mice related to depression in humans. So normally mice are very social. If we let them explore an empty arena, they wander around fine. And then if we put a novel mouse they've never met before in this interaction zone, a little cage, the novel mouse can't get out, but the other mouse can explore this and decide whether it wants to interact or not. Control mice that have never been bullied, very social, spend a lot of time right up nose to nose with this novel mouse that it's never met. However, animals that have gone through this bullying stress are very socially avoidant. They will go and explore a little bit. For the most part, they know this is maybe not a great social situation, and they're much more socially avoidant. So this is the assay, one of the main assays we use to try to understand how stressed the mouse is after this bully stress. So what happens when they've experienced early life stress alone, no adult stress? When we measure their social interaction, we actually don't find a very large effect of the early life stress by itself. If we look at these heat maps of their behavior, you can see, yeah, they spend some time in the corners, but actually the early life stress mice spend a lot of time investigating. So early life stress by itself doesn't seem to have a big impact on this social behavior. However, the mice that have experienced early life stress and the second hit of stress in adulthood, particularly if the early life stress was in this later postnatal period, are much more socially avoidant. They have reduced social interaction. You can even see it in the heat maps of their behavior. They're sort of pacing around the outside, but really staying far away from the novel social targets. They've overgeneralized this anxiety response to the social situation. So we think that this gives us evidence of a potential sensitive period in rodent development, at least, for early life stress to increase risk for depression-like behavior later in life. But sort of when is this in terms of human brain development is always the question. And this is not a question we actually have a concrete answer to. So I like this slide. It's fairly comprehensive, but different brain regions develop at different speeds, and there might be different organizational mapping within different regions from humans to rodents. So it might be that a prefrontal cortex matures at sort of the same rate in humans and rodents, but maybe reward circuitry is a little bit different. But we think our best guess, is that, oh, we don't like that. Okay. Our best guess is that the period where we're stressing our mice, right over here, after postnatal day 10, but before postnatal day 20, really is much more aligned with early childhood and the pre-adolescent period in humans. And it turns out that the first week of a mouse's life is a lot more like the last trimester of human gestation. So we do think that our stress-sensitive period broadly maps onto early childhood. Okay, so how can we use this mouse model to understand changes in the brain? And now focusing on changes at the level of cellular activity. And one of the questions that I'm really interested in is we see these latent changes in behavior. There's not a lot of changes right off the bat. We've actually looked at a lot of different behaviors, not just the social behavior. And none of them are different after early life stress alone. They only have this latent response to a second hit of stress later in life. So how are we sort of priming this response to a future stress? And again, I'm focused on reward circuitry. So in the human brain, this is going to include the ventral tegmental area and its projections. This is a dopaminergic region of the brain, and it sends a lot of projections to the ventral striatum. In a mouse brain looks a little different. The VTA sends projections to what we call the nucleus accumbens over here. So these are the regions that I'm focused on. And these make up the main reward circuitry in the brain. And once again, we see evidence from literature that reward circuitry is involved in stress and depression and responsive to early life stress. And I'd already sort of shown you some of these things. We also see large changes in gene expression and epigenetic regulation of genes. And this is a lot of work that I've done. Yep. Yeah, sure. I'll explain more when I get there, but central dogma of biology is that we have DNA. It's made into RNA, and the RNA is made into protein. So the genes don't change. The sequence of our DNA never changes. What's really interesting to consider is sort of the rheostat or the volume dial on our DNA, how it's being made into RNA. Is it being made into a lot of RNA or not very much RNA? And so that's the gene expression, the level of the genes that are being produced. And how do we change that volume dial is actually the structure, the three-dimensional structure of DNA and sort of how we decorate it. And I'll talk about that when I get to more epigenetics. Thank you for pausing me there. Okay, so the first question we wanted to ask is, are cells that are responsive to the experience of early life stress reactivated during experience of stress later in life? It seems like a basic question, but it actually turns out this is a challenging question to answer. Partly because we have a lot of tools in neuroscience to look at cells that are active right now, but really no tools in neuroscience until very recently to look at what cells were active a while ago. So we've used some fancy mice to solve this problem. We're actually able to, for the aficionados, I'll put up our breeding scheme, but don't worry about this mostly. We're able to tag cells that are responsive to a specific experience and have that tag stay in the cell for the rest of the animal's life. So what does that look like? If we go in, oh, a little bright. If we go in, the dots in blue here, which are a little hard to see, are all the cells in a brain region. The dots in red are all of the neurons. So the brain is made up of neurons and non-neurons. So we can see all the cells and all the neurons. And then our fancy tag that we're able to capture cells only active during a specific experience puts a nice little green fluorescent protein ring around the nucleus of these cells. So we can go back at any time in the animal's lifespan and look and see which cells were previously activated during this experience. And so we've decided to tag cells that are activated during experience of early life stress and during control experiences. So the first question, are cells active during early life stress? Are those reactivated during adult stress? And we can see, it is a little hard to see. Hopefully you can see that some of these cells that are active during early life stress also have activity markers, a different kind, during, oops, so sorry. That's usually when I go home to pick up my kid. Also have this activity marker in response to adult stress later. And you can see a lot of overlap of these cell populations. And this is in the adult brain. So this is in one of the regions that I've highlighted, the ventral tegmental area, this dopamine-rich region. We also see evidence of that in the nucleus accumbens, where these dopamine neurons project to. Again, a little challenging, but you can sort of see where these arrows are pointing to. This is a marker of adult stress, a marker of early life stress, and they're overlapping here. So now the challenge is, how much overlap are we seeing? Focusing on the nucleus accumbens, this sort of projection region, we first counted how many cells are activated during our early life stress versus the control experience that we gave them. And there's no differences. This actually turns out to be really good. So the control experience is activating roughly the same number of neurons as our early life stress in this brain region. But we think that these are different cell populations, different types of cells that are responsive to these different types of experiences. Next, we wanted to know, how active this brain region is in response to the adult stress. And it turns out that much like our behavior, there's more activity if an animal had experienced, oh, I'm going to take you back. This is much prettier. Here's the good one. Okay, yeah, now you can see this overlap here. Okay, double cells. This was a cool moment in the lab. Okay, we can see that much like the behavior, animals that experienced both early life stress and this adult bullying stress have higher levels of cells that are activated overall. And we see that this overlap is also increased specifically in the animals that experience the 2 hits of stress. So the 2 hits of stress together, these cells are double labeled, so they're activated at higher rates. Looking at this just one other way, asking, are early life stress activated cells? So now we're only looking at that population. Are they predominantly or preferentially reactivated by adult stress? And we actually see evidence that this is true. So this is pretty exciting. This is not the case across all brain regions, but it does give us some evidence that cells that are responsive to early life stress are the ones contributing or being reactivated by adult stress. So this was cool, but we wanted to know, is that important for their behavior? So we then asked whether we could actually quiet the activity of these cells to promote resilience in the face of future stress. So for the neuroscience students in the audience, we use a DREAD approach. We expressed special receptors in these cells that allowed us to tamp down on their activity and quiet that activity. Here's our breeding scheme for those who want to know it. So what did we actually do? We had to do some surgeries on these animals when they were very young. We put them through early life stress, and we tagged our cells, either in response to early life stress or in response to just an enrichment experience. We then looked at their behavior and then put all of our mice through bullying stress while they either got the ligand that activated this receptor, the special receptor that allowed us to quiet the cells, or just a placebo, the vehicle. Looked at their behavior again, and then switched all the animals. So actually all of our animals went through the bully stress twice. One of the experiences they had their cells quieted during the experience, and one they were allowed to experience it in full, all the cells active. Then we tested their behavior again. So what did we see? Animals that had not experienced any stress yet had similar levels of this social avoidance behavior. Animals that only received the placebo or the vehicle, we see the early life stress, we replicate the effect, it does induce the social avoidance. But when we quiet the cells during that stress experience, we're able to rescue this depression-like or social avoidance phenotype. When we look at which animals we might characterize as susceptible or resilient or indifferent, giving them a bit of a buffer zone, we again see that early life stress alone produces a lot of animals we would consider susceptible to the bully stress. But when we quiet the nucleus accumbens or early life stress activated cells, we promote resilience in these animals. So we bring down the level of susceptible animals. And this is really cool, that we could actually change activity in the brain and make them more resilient to that stress later in life. So we think this gives us evidence that the nucleus accumbens is an important region for experiencing this sensitized response to future stress. We then wanted to know what's happening in these cells, what's happening at the molecular level. So we asked whether early life stress primed activation in response to future stress. Now we're asking whether early life stress primes the mRNA that's being made in the brain. So how did we assay this? We took our brains, we took these little tiny pieces of tissue from the brain regions we're interested in, and instead of looking at one gene at a time, we actually decided to be agnostic, and we wanted to look at all of the genes all at once. So we used something called RNA sequencing. And then the fun part's in the analysis here. So at a very high level, we can just look at the number of genes that are changed in response to each of these different stresses or combinations of stresses. We can see that in both male and in female mice. Certainly, there are a lot of genes that are altered after early life stress alone. There are a lot of genes that are altered after adult stress alone, unsurprising. But we're really excited to see a lot of genes altered by the combination of early life and adult stress. And this wasn't just a gene that had changed a little bit and then changed more after the second hit. These are totally unique genes that we saw to be altered, which again gives us evidence that, that there might be this priming of this latent gene expression response to the future stress. So we wanted to dive into this a little bit more and look for specific patterns of genes where they were not altered after early life stress, not altered after adult stress, but were either forced up or turned off after the combination of stresses. And we consider this sort of a priming pattern. And in fact, we see a lot of different genes in different brain regions that follow this priming pattern. And I've just highlighted some for those who might be interested. So we think that this gives us evidence that there is a gene expression response that's latent, that sort of mirrors this latent behavior change that we see. We next wanted to know, how is this possible? Why are these behavior and gene expression changes latent, and how does this priming occur? And to try to understand this is when we turn to how gene expression is regulated by the epigenome. And so in order to make sure we're all on the same page about what the epigenome is, I want to back up and take us back to some cell biology. Hopefully everyone will stay with me. But it turns out that the three-dimensional structure of DNA is really interesting and important. So we don't just have a couple genes and they're always on. We have a lot of genes, and sometimes they're on in some cells and sometimes they're off. And in other cell types, they're on or off. And the DNA is organized in a way to allow different expression at different times. So how is it organized? It's organized in a nucleus in something called chromatin. So this is our telephone cords, for everyone who remembers corded telephones. It's really tightly wrapped and wound in order to stay organized in the cell nucleus. So we have this closed chromatin. It's DNA wrapped around proteins, and that's how it's very compact in the cell. When it's really, really tightly wrapped like this, we call it closed chromatin, but it can actually be unwrapped and more open. We're calling this open chromatin. And when it's open, then we can actually make RNA from these genes. Now, when I was in high school, we thought that there was either genes or junk DNA, but now we know that these regions of DNA that aren't just for genes are actually very important. It's not just junk, and they have regulatory functions. And so I'm going to teach you the names of these regions for those who are not molecular biologists. If we have our gene that needs to be made into RNA, the region right next to it that helps promote expression of the gene is called the promoter. Makes sense. We know a lot about promoters, and we're starting to learn more about these really far away regions called enhancers that, as you might guess, also enhance gene expression. So aptly named both of these regions. And it turns out that this looping that occurs where enhancers contact promoters is really interesting and important for regulating gene expression ultimately. So as I mentioned, the epigenome helps keep everything organized, and it's different from thinking about changes in the genome. Changes in the genome are when you actually change the sequence. Changes in the epigenome is all about changing this three-dimensional packaging. And it turns out that while the sequence of our genes cannot be changed, we can change the epigenome just by the experiences that we live through. Okay, so one more characterization of this. We have this closed chromatin. The DNA and the proteins are wrapped around and really tightly wound. We can also have this open chromatin conformation. And if it's open, but the enhancer and the promoter are not touching, we call this primed. So it's able to respond to cues that might express a gene, but it's not actively expressing any genes. This is marked by a couple particular chemical modifications that we're interested in. And then finally, if we want to actually activate our gene expression, we need to bring the enhancer to the promoter, and then we can express our gene. Okay, now it turns out that it's not just DNA wrapped around chromatin. This chromatin has little tails sticking out, and these tails can be decorated, which helps wind or unwind it. And we happen to be very interested in these decorations or histone modifications. And if we look at all the decorations on the chromatin, we can see actually the early life stress changes these decorations in a way that we think changes how open or closed the chromatin is itself. My lab also wanted to look directly at, well, is chromatin more open? Is it in this primed, is it in this prime, open but not active state that we might hypothesize? So we use something called attack sequencing, which literally is an assay to look for open chromatin. And we ordered we sequenced the entire genome in the brain regions that we're interested in. It's reward circuitry. We looked at what regions were open, what regions were closed. Closed is in red, open is in blue. And in fact, we see that early life stress promotes more open chromatin, which gives us some evidence of potentially the genome being more primed to respond to future experiences. We can look at this in another way. This is called a volcano plot. You can see sort of spewing data out from the center here. Points this direction are more open with early life stress. Points this direction are more closed early life stress, and this is how sure we are of that. And we can see that there's 700 regions that are more open after early life stress in this brain reward circuitry, and only 400 regions that are more closed. Diving into this at one more level, we can look whether these open chromatin regions are in particular types of or locations in the genome. And we wanted to know, are they in promoters or are they in enhancers? And it turns out that we have more open chromatin in these enhancer regions that I told you about, which are the things that can be sort of primed for future gene expression activity, but not necessarily active right now. So we think that this is a mechanism that early life stress is using to change the epigenome, to change the structure of DNA in our cells, to make it more reactive to future stimuli, such as stress. So if it's open a little bit but not active, then stress can come in and sort of hyperactivate it more easily. But we weren't satisfied there. Next we wanted to know, well, is this important? Can we artificially prime chromatin? This is one of the marks that I glossed over, but this is a marker of open chromatin, and we can actually force it. We developed a tool to force chromatin to be more open. So we overexpress this enzyme that establishes one of the marks of open chromatin to try to open up the chromatin itself. We did surgeries in our mice to express this, or the control vector, and then we put animals through this bullying stress in adulthood. And when we look at their behavior, we see that overexpression or opening up the chromatin by itself in the absence of stress doesn't really do anything to their behavior. But if you have open chromatin and experience stress in adulthood, we see more depression-like behavior. So artificially opening up or priming the epigenome in these animals is sufficient to make them more sensitive and more responsive to adult stress later in life. But it is a big jump to go from this tiny molecular level to behavior. So we also wanted to know, is their gene expression also primed? So I told you about volcano plots already, but we see that when we prime the epigenome early in development, not only do we see hypersensitivity to adult stress at the behavioral level, we also see a hypersensitivity to genes, to gene expression, and the amount of RNA that's also responsive to the adult stress here. For comparison, if we hadn't primed the epigenome, we'd see mostly genes going down in response to stress in this brain region. So again, we think that this gives us a lot of evidence to show that priming the epigenome and opening chromatin is sufficient to sensitize animals to future stress later in life. Okay, so this is all about what we find after early life stress, but how do we promote resilience in these animals? So we have our standard root animals and our early life stress animals. This is how we sort of think that the chromatin is structured. We have a stable chromatin conformation in the standard root animals and a more reactive, hypersensitive chromatin conformation in these early life stress animals. And we want to take this state and put it back to the state. So right now my lab is developing some CRISPR tools. I think most people have heard about CRISPR in the news at the very least. We're harnessing this technology, changing it a little bit, and we're putting it back into the brains of animals to promote closed chromatin and test whether this is, whether closing the chromatin actually promotes resilience. So So the pros of this approach are that it's highly specific. We could target very specific locations in the genome to close it back up. The cons is perhaps it's too specific and it's really difficult to bring to a human population, or at least we're very far away from that being useful in humans yet. So in another study, we used a small molecule inhibitor of a different chromatin modification that we see changed after early life stress, this drug penemetastat. And we can inject this systemically into animals, just regular. Once again, we see the early life stress changes their depression-like behavior, increases social avoidance. And actually, if we give this epigenome in an inhibitor, we can reverse these changes in the epigenome and also reverse their depression-like behavior. So the pros is this is a drug and it can be useful in human populations potentially. The cons is if we're injecting it into the whole body, this is going to have a lot of off-target effects. So the challenge for neuroscience always and for developing translational approaches from mice is to hit that sweet spot of specificity, which I think we're still working towards. Okay, so some conclusions from our lab. Early life stress sensitizes humans and animals to future stress. It makes cells and brain reward regions more reactive to future stress. And quieting this activity can promote resilience. We also see these latent changes in RNA following early life stress. And we see that early life stress primes the epigenome, or the structure of DNA, to be more reactive to future stress, which we've shown is sufficient to facilitate this hyper response. And we're now working to see if reversing these changes can promote resilience. Okay, so focusing on resilience, what do we know? What lessons can we take away from molecular studies? And then lessons from psychology and neuroscience more broadly. The takeaway point that I want everyone to know is that resilience is an active process. It's not simply a reversal of changes with susceptibility, but it's its own active process. So some literature has suggested, this is a recent paper where researchers used MRI to look at a lot of different brain regions and look at the connections between these brain regions. This is in a control subject here. And in children who had been maltreated and had symptoms of anxiety and depression, they see this hyperconnectivity between this one frontal region and other regions of the brain. And surprisingly to these researchers, children who had experienced maltreatment but showed behavioral resilience, a lack of depression and anxiety, actually had reduced connectivity. And this seems like an active process. It wasn't just a reversal of the changes in symptomatic children, because this is actually a lot lower connectivity than controlled. But it's something, refining these connections in the brain as an active process. Molecular studies also give us evidence that resilience is an active, independent process. When we look at gene expression levels, again, if mRNA is up, it's in yellow. If it's going down, it's in blue across the whole genome. We can see a lot of similarities in animals that experienced stress and ended up behaviorally resilient versus behaviorally susceptible. So there's obviously a huge stress component. But there were actually more gene expression changes in animals that remained resilient, which is really interesting. We can use this information to understand also how individuals respond to antidepressant treatment. And it turns out that successful response to treatment is a combination of reversing susceptible genes and inducing resilient genes. So I'm not showing all of that, but the takeaway message from this paper down here is that response to antidepressant treatment is both. It also has to include an induction of these resilient gene expression changes. Okay, and so finally, what can psychology and neuroscience teach us about promoting resilience in children following stress? And the three takeaways that I'll highlight are just to be present for your kids or kids in your community, to talk to kids, and then there's some recent very interesting evidence people may have seen attention to that money solves problems. So more money, less problems, in fact. So be present and potentially involve community. It turns out that parents and caregivers buffer a child's stress response, at least until adolescence. Then they're on their own. Then they're more peer motivated. But in earlier childhood, mere presence of a parent can actually buffer not just hormonal stress response, but stress response that we see in the brain at the level of the amygdala. And this is one of the reasons why we think child separation in the study that I highlighted earlier is so detrimental to children, because the absence of that parent present means a child's stress response is going to go unchecked. And it also takes a village. So if you have more close caregivers and friends and family and coaches in your community, you have more chances to buffer stress and promote resilience and other active coping stress responses. And we have a lot of lessons from Head Start and early Head Start that's saying promoting family engagement, just continual engagement with the children, really does reverse a lot of the detrimental effects of poverty and other adversity. Talking to children is important. This brain region highlighted is actually in the language center of the brain. This is one of the regions responsive to childhood adversity that might be important in a number of different dimensions. But talking and reading obviously helps build language and these language centers in the brain, which might promote resilience overall. Naming things helps us understand them. And talking about stress helps children process their stress feelings with us. It gives them examples of positive coping. Reading to children has been very deeply studied and definitely part of what makes children resilient and able to reach educational milestones, and with that comes a lot of other positive coping and health. And then finally, there's recent evidence that money, in fact, does solve problems. So this is research out of Kim Noble's lab that just came out last month, in fact. But my second son was able to participate in a study from that same lab. And this study looked at brain signals through EEG. This is the EEG cap that he was wearing when he was just 1 month old in the study. And they found that predictable monthly unconditional cash, they gave all the participants $300 or I think $20 a month, given to low-income families, directly impacted the brain activity of children. I will disclose that my son was not participating in that particular study. And one of the reasons this is important is because it shows that income or poverty on the flip side is actually causal for changes in brain development. And interestingly, in the high cash brain patterns were very similar to what we already know about brain patterns, similar to higher language, cognitive, and social emotional measures later in childhood and adolescence. Once again, showing us that resilience is an active process. I think we're perfectly on time, so thank you to all of you. Thank you to my lab and our collaborators. And for those who are interested in helping children, not necessarily through research, but if you have means, there's a lot of important foundations working with children at different levels, either internationally or nationally. So these are great organizations to find, get involved in, or donate to if you can. And thank you. Yeah, does anyone have questions? Great question. They are mostly not, in fact. It seems like, well, this is, I can talk about this for hours actually. It seems like the environment that you are raised in, reared in, is the environment that changes your epigenome. So in animal studies, when we adopt into a different early environment, it's not the environment that your parent experienced, it's the environment that you yourself are reared in that seems to program your epigenome. So they were looking at the rhythmicity of these different brain signals in the EEG signals, and they found changes in activity between children who were in the low cash and the high cash groups. Everyone got some money, but the higher income supplement group showed different patterns of brain activity that are related to what people had seen in other studies of patterns associated with higher language, cognitive, social, emotional skills. So they were just looking at these EEG sort of cyclic signals in the brain. Yeah. Yeah. Yes, absolutely. So the regions we're focused on are this reward circuitry. They are involved in stress and depression, but they're classically known to be involved in rewards, such as response to drugs of abuse. And there's obviously a huge literature as well about increased risk of substance use disorder following traumatic childhood events. Yes, we absolutely think so. So one of the things that I am looking forward to doing in the future studies in our group, we've looked at how early life stress primes response to future stress, but we also think that these same mechanisms are involved in response to future something else, future drug exposure, possibly future enrichment. So this period is very plastic, and we think that we can also potentially intervene in a positive way, just at the behavioral level. In rodent studies, this looks like giving them wheels to run on and things to play with, but in enriching environments, which we see from the early Head Start programs as well, to harness this plasticity in a positive way. But yes, the same changes that are happening that are sort of promoting stress susceptibility are probably also the ones that we need to harness to promote resilience. Yeah, so in our double tagging study, we looked at a lot of different brain regions. We looked at accumbens, ventral tegmental area, prefrontal cortex, and basolateral amygdala. We see a robust response to adult stress in the basolateral amygdala, but no synergistic interaction between early life and adult stress, which we were surprised about because so much of the literature is focused on amygdala to PFC. So that's just not the mechanism there. It's possible that we're still seeing developmental curves shifted and sort of that uncoupling of where the brain should be developmentally versus the sensory experiences like parental care that it's getting are contributing, but we don't see a hyperreactivity there. Well, it integrates a lot of signals. Nucleus accumbens receives input from dopamine neurons in the ventral tegmental area and glutamatergic neurons from all over the brain, hippocampus, prefrontal cortex, amygdala, et cetera, hypothalamus. Other studies have shown that in adult stress, when we're looking at susceptibility and resilience, there's a strengthening and weakening of connections that the accumbens is integrating, where susceptibility is associated with stronger prefrontal cortex connections to the accumbens, and resilience is associated more with weak hippocampal input, which I think is funny because it makes me think that my own stress resilience may be due to poor memory. But because it's an integrator, where I started going with this is it might just be not able to sort out all of the inputs coming in. If it's just if it's hyper responsive to everything, I think it's probably just not able to detect the focus, the center surround kind of idea, and focus on what might be important in the moment. And it's getting sort of overwhelmed by other types of input, such as emotional valence or something like that. Yeah. So there are a lot of studies that find effects of early life stress by itself, particularly, well, most of the literature does the stress in the sort of earlier period that I didn't find effective in our hands. They're mostly focused on changes in exploratory activity, so open field tests for those who know about rodent behavior tests. We do see some changes after early life stress alone in exploratory activity, certainly, but not sort of not a dual response to the adult stress. There might be a lot of things going on. A lot of rodent researchers buy, this is going to weird details, buy animals from breeders while they're pregnant, so they can get going right away in the animal studies once the animals arrive. But shipping is a stress for rodents, so we think that by the time they get to their early life stress, that's actually already a second hit. We're breeding our animals in-house, and so we're controlling that environment a lot tighter, and we think that our second hit really is later in life. Thank you. Thank you all.